Hello and welcome to DNA Today. I am Kira Deneen, your host. DNA Today informs you on what's happening in the genetic world. The show is packed with new stories and lessons, all to keep you updated and educated on what's happening in the world of genetics. And it's been a busy week in genetics, at least in Connecticut. The grand opening of Jackson Laboratories in Farmington, Connecticut was this past Wednesday on October 8th. And I was lucky enough to be able to attend. I got to miss my genetics lecture for it, so a little bit of a trade-off, but, learned a lot at the opening. And I met so many great people in the field and a lot of UConn people and professors, and it was really a fantastic experience just to hear from Nobel laureates or Nobel Prize recipients on their work and a lot of other people. There were 8 presentations on research. Most of them were like half hour, 40 minutes. And so it was really a packed day and awesome opportunities for networking, as I said, and it was just, excitement was in the air, and it was really cool just to be able to hear of it from, when they decided to open the Farmington location to actually being able to walk around and, see the location and everything at UConn Health Center. So it was a very exciting day for me, as well as Jackson Laboratories and a lot of other genetic-related companies that were there, represented by people, and it really was an awesome day. So I just kind of wanted to recap, what I experienced and also what I learned, and I'll start out with a few things that I learned about Jax, or for those of you that don't remember, I have referred to Jackson Labs before on the show, in my anticipation of attending the grand opening. But for those of you that don't remember, Jackson Labs is an independent nonprofit biomedical research institution, and they focus on genetics and genetic research, especially in terms of cancer. And they have a partnership with UConn, as it is on the campus of UConn Health Center in Farmington. And they also have a partnership with Yale University. So they may be kind of small compared to other genetic companies and genetic research places, but they definitely are rapidly expanding, as I've said. And their mission is we discover precise genomic solutions for disease and empower the global biomedical community in shared quest to improve human health. And their kind of tagline you see on everything is leading the search for tomorrow's cures, which I like. It's nice, simple, streamlined, and little excitement in there. They have a lot of research funding. Their total federal research grants awarded to date for Jackson Genomic Medicine, so that's just the Farmington location, for their scientists is $14.2 million. So that's quite a bit. They definitely are very prevalent in this area and in genetics. The total staff as of September 30th of 2014 is 1,637 employees, 1,500 of which are in Bar Harbor, Maine. That's their headquarters. And beautiful area up there, by the way. But so most of the people are in Bar Harbor, and some obviously are in Farmington now, that they're hiring people for Farmington, and also their location in Sacramento, California. And what's really cool about Jackson Labs, which a lot of people don't know, it's the world's source for the more than 7,000 strains of genetically defined mice and home of the mouse genomic informatics and mouse phenome database. So they are really the place for mice. And as you guys probably can think, like when there's a lot of genetic research, a lot of companies use mice because they really are very similar to humans in terms of how much of our genetics are shared. Of our genome that's similar, and also they're small enough, and their reproduction cycle is fast, and so all around they're really great to do research on. And Jack's Genomic Medicine, again, that's the name of the Farmington location, they're going to include 17 wet bio labs and 17 dry computational labs. And the building, again, is beautiful with, open, light feeling, light wood and everything. And the auditorium seats 200 with 2 additional large conference rooms. And when they were presenting, they had, 200 people were in the auditorium, and the overflow was in the conference rooms. And it was really cool. They had they were filming the presenter, and you could also see people in the room, too, on the TVs, and they could see you in the other rooms. So if you asked a question, you could ask questions in the conference rooms, and everyone could see on a TV in the main room. So it was really cool the way they set it up and everything. It was really open and friendly and everything. And there's even an employee fitness center there, and didn't get to see it, but I'm sure it's really nice. And there's a casual sitting area on the second floor, referred to as the pub or the bar, and which is also really nice. Everything is brand new because it just opened. And the third and fourth floors are predominantly research labs and faculty offices. And the grand opening caps 669 days of construction since December 2012. So they definitely made progress. And I was reading a lot of the materials of just the head people in Jackson and head construction workers talking about how they really did stay with their timeline of making sure that they're going with, all right, we're staying up to date on this phase of the construction and the next phase. And even with those freak snowstorms, back in, I think it's 2012, 2013. Maybe it's 2013. Yeah, that sounds about right. But yeah, so they really kept up to date and, right to the day. And as I mentioned, something to note is that Jackson has 2 other locations in Sacramento, California, and their headquarters in Bar Harbor, Maine. So who knows, maybe there'll be a 4th coming down the line. But right now, Farmington is the new addition to Jackson Laboratories. And the grand opening, the main themes of the symposium included clinical genomics, modeling human diseases, and genomic technologies. So all of the talks kind of centered around one of these or multiple of these, so that those were kind of the focuses of the day. And the beginning of the conference, they talked about hot areas in genomic medicine. And this kind of means where the money's being invested in genomic medicine. So I'll just kind of go through some of them and talk about some things that they mentioned. The first one was cancer genomics. Obviously, Jackson Lab is focused on cancer research and looking at how genetics play a key role in cancer. Another one was pharmacocytogenomics. And the third was genomic medicine test drive program. So an example of this would be clinical sequencing exploratory research. And the fourth one was really interesting. This was kind of geared more towards the general audience that was there at Jackson, which most people there were scientists or researchers, or genetic counselors, or really people in the field practicing. But there were some people like me and other students there that, some of it kind of went over our heads. But it was interesting to hear when they talked about prenatal and newborn genomic sequencing. And the idea here is that every child born has their genome sequenced right away. And it's an interesting concept that may actually become a reality someday. So by sequencing genomes of newborns, we could screen for more diseases, and it become more of a standard of practice that when a baby's born, then their genome is sequenced right away. And this way, a patient's sequenced genome can be referred to throughout medical treatment of their whole life, that they can just keep going back to it and saying, oh, do you have this mutation, or do you have these genes that are associated with this, et cetera, et cetera. And there's also a lot of ethical issues that go with this, obviously. How would the data be stored securely while still being accessible by doctors and other healthcare professionals? And also, if people want to know whether genetic diseases, if they want to know if they are susceptible to genetic diseases, because some people would rather not know. They'd rather kind of just go live out their life, especially if a disease, maybe there's some treatment, but there's no cure, then for them it's kind of like, well, there's nothing I can do about it anyway. I don't really want to know until it happens to me. So while other people, maybe they want to know and say, no, I want to plan my life. I want to know what's coming. I want to be prepared. So it really depends on the person. It's a very personal decision. We've talked about that before. And so if once a patient's genome is sequenced, it's tricky for doctors if they do see it, like if they see like, oh, they say test positive for the BRCA2 gene, which we're going to talk about later. Say they test positive for the BRCA2 gene, then, does the person want to know that? Do they not want to know that? And then if the doctor sees it in front of them, maybe they'll mention it by accident. So there's a lot of things that are tricky about this, and a lot of ethical decisions that go into this. Now, some people argue this would be more beneficial, as it could streamline the process of identifying genetic conditions early on. And so there's kind of this toss-up, is this going to become a standard of practice? Are more and more people going to be doing this? Is this going to become what everybody does? So that was really interesting discussion, just to hear how fast things are moving in genetics and how the public is becoming way more aware of things happening in genetics. And, obviously that's my aim here, for you guys to understand more of what's happening in genetics and everything and stay updated. But it's very exciting to hear that places like Jackson that really focus on as much research as education. They have a lot of symposiums and other conferences that I'll mention towards the end of this, just if people are interested in attending. But anyway, going back to this, the clinical genomic information systems is another hot field mentioned. And the fourth here, I believe, is the ultra-rare genetic diseases. Now, these are not rare diseases, but ultra rare. So kind of even further, that next level. And through new technologies, they're being identified in cases that were previously thought to be unknown. So if someone has what we think might be a genetic condition, there's kind of no answers for them. But now, through these new technologies that are being developed, these ultra-rare diseases, ultra-rare genetic diseases, are easier to identify and say, we think you have this. There's a mutation in this gene or these genes or something like that. And so that's really exciting to be able to be finding these. So there's maybe a couple cases in the world or something like that where these are being identified. And sometimes, I don't remember who said it, but someone that was presenting mentioned that sometimes having an answer for them, this is what's wrong with you, even though there is no, here's the cure, here's the treatment, just knowing is sometimes a little bit more relieving than it's like, all right, we know what's wrong with me kind of thing, even if you can't do anything about it. So there's that level of kind of just feeling better about it. So, and obviously once we identify these, maybe we can then find treatments and cures and everything. So you got to start somewhere. And just to mention here, there are two main types of genetic diseases, and one is rare, simple, monogenic Mendelian. So that's one kind of set. And so examples of these are cystic fibrosis and Huntington's disease, both of which I talked about previously on different episodes. But these are diseases that are due to simple mutations. So Huntington's disease, we talked about how that is, I'm going on a limb here and say I think that was chromosome 4, that there's one of the genes is mutated, and that's how Huntington's disease happens. And so it's because of just one straightforward kind of change that it occurs. But it's rare. It's not, it doesn't happen to a lot of people, but we do know of it. It's not ultra rare where you've never heard of it, but you've heard of cystic fibrosis, you've heard of Huntington's disease, and other Mendelian rare simple disorders. And then the other main type is common, complex, multigenic, non-Mendelian. So multigenic, that's meaning multiple genes are involved in the disorder or condition. So example of this is autism, where there are multiple factors that cause a condition, and we don't know. There obviously could be more than just genetic things that are making things like autism occur. So that one is way more complex, and so treatments and cures for these are little bit tougher. And the last hot topic in genetics that they talked about, or genomic medicine that they talked about, is generating a human genome sequence. And this one still blows my mind. It's becoming trivial at this point, just to be like, run the human genome sequence and just figure that out. And it only costs around $1,000 today to sequence a human genome. You can get it done for $1,000. And it used to cost an outstanding $100 million only 14 years ago. $100 million 14 years ago. I know I've talked about this before, but it's still blowing my mind that now it's only $1,000, and before it was $100 million only 14 years ago, less than 2 decades. So it just shows how far we're coming and how fast the technologies are advancing, and how much that means that 10 years from now, who's to know where we'll be. And that's another thing, that whenever they were referring to research in the future of, plans, what we'll do, they're like, but all this could change because how fast technology is advancing, how much we're learning. And so what you think would be a 10-year plan, maybe 2 years from now, it's not accurate anymore. It's not really on point. So it's so exciting to just hear of so much research happening and how much it's really changing. Another thing they mentioned, a little tidbit, was the relevance of genomics. And they kind of mentioned 3 main areas. Biomedical research, there is huge focus on genomics, which is kind of understandable. We've been talking about that a lot, and the biomedical industry in Connecticut alone is expanding. But also in healthcare professions, healthcare professionals are starting to see the potential, especially after the Human Genome Project. Once that was done, that we have a full draft, not even a draft, like a finalized version of the human genome through the Human Genome Project. Now healthcare professionals are kind of looking at that as another source to see, can we use this to identify things? Can we use this to think about treatments and things like that? And the third aspect, the third area, is patients. Patients hear about, there's now commercials on TV for genomic medicine and genomic research. And so now they're asking their doctors, hey, do you know about, should I get my genome sequence? Should I do this? And is this going to help me out in any way? And of course, from the patients, the friends and family of the patients, their loved ones, that they're invested in the patient as well. So it's becoming way more relevant just in the mainstream, that it's kind of breaking out of its realm of just staying in the science and genetics field of people in the field talking about it to now, public wide. And, there's been Time, the magazine Time has had issues of talking about genetics. And now, as I said, there's commercials on TV. So it really is becoming more of a mainstream thing, and more people know more of the basics of genetics. And there are 4 presentations of research, there are 14 presentations of research, and I'm going to go over the top 3. These are the ones that people that were Nobel Prize winner recipients talked about, because obviously they were kind of the distinguished speakers out of everyone. The first one is Dr. Mario Cappucci, and he is a distinguished professor of human genetics and biology at the University of Utah. And in 2007, he won the Nobel Prize in physiology or medicine for discovering a method to create mice in which a specific gene is turned off. And so this is known, like the gene being turned off, that's known as knockout mice. And so he presented this in his lecture titled Gene Targeting into the 21st Century, Mouse Models of Human Disease from Cancer to Neuropsychiatric Disorders. And so basically this is a mouse created by genetic engineering and in vitro fertilization, in which a particular gene has been turned off. So he was able to, along with colleagues, he was able to figure out how to turn specific genes on and off. And he did a lot of research with the Hox gene family, which is a gene associated with controlling embryonic development of mitocellular animals of the orientation of the body. So where the arms and legs location are and how the body knows where to grow those. And so that's the Hox gene family. And so he was doing research on how that works and how that works in the orientation of the body. The next presenter, not the next, but another presenter was Dr. Bruce Butler, and he is the Regeneral Professor Director of UT Southwestern Medical Center. And in 2011, he won the Nobel Prize in, same thing, physiology or medicine, because of his discoveries concerning the activation of innate immunity. So his talk was Forward Genetic Discovery in Mouse and Man, and his lab is the only lab where the phenotype or physical traits is recorded, and the cause of the phenotype is known within an hour or rejected as non-genetic. So he's able to take, say, a mouse and say he doesn't know what the physical traits are. And I'm not just talking about fur color. I'm talking about things, on the inside that you wouldn't be able to see, certain organs, certain proteins, things like that. And this is able to be recorded, and the cause of the phenotype is known within an hour. And if it's not known, if it doesn't come up as in the system, it's rejected as non-genetic. And this is because of their database and also the technology that they use to be able to do this. And this technology can operate even if the phenotype is not visible. So as I said, it doesn't have to be like fur color, like that's kind of obvious. It can be something that's non-visible. And genes can have different levels of production. So even the same gene between two people can have producing different amounts of the protein. And so this concept is known as penetrance. And so 100% penetrance is not needed for the technology to operate. So you could have a lower level of penetrance, and so that's when, the gene isn't producing as much protein compared to other times. And so it's not even needed for that. So it's really advanced technology and really impressive. I was really intrigued by his presentation on all of that. The third presenter I'm going to talk about is Dr. Jack Sidstak, and he's Massachusetts General Hospital and Howard Hughes Medical Institute. And in 2009, he won the Nobel Prize in physiology or medicine. And his talk was Adventures in Science from Telomeres to Protocells. And he's credited with the construction of the world's first yeast artificial chromosome. So he actually constructed a chromosome artificially in yeast. And this discovery assisted scientists to map out the location of genes in mammals and to develop technologies for manipulating genes, because obviously he created one, so he's pretty good at manipulating genes. He has that ability. And it can help also to map out the location of genes, because in order to create the chromosome, he must have had to understand where the genes were map-wise and the location of everything. And he's done quite a lot. His work has also contributed to the understanding of the process of chromosomal rearrangement, which is the shuffling of genetic information during meiosis, the process of making sex cells so that you don't have all kids that look the same. And this occurs to enhance genetic variation again, so you don't have, all the same looking kids. Got to tell them apart. And a good portion of his lecture was also talking about telomeres. And these are regions of repetitive bases, ATCG, like that, it'd be the same one repeating, at each end of the chromosomes. And it protects the chromosomes from being degraded or fused with other chromosomes, so they're not combining with others. And I kind of like comparing it to the end part of your shoe, the aglet, that plastic part. It's kind of like that because it prevents the shoelace from unraveling. So the telomere is that cap at the end that's preventing the chromosome from degrading or combining with others, kind of keeping it stable. So those were kind of the highlights that I got out of the grand opening. And just to mention, there were a lot of other undergrad and grad students in attendance. I even saw one high schooler on the list. So I was very impressed by that. And many people from UConn Health Center, Connecticut Center Medical Center, National Human Genome Research Institute, a variety of universities, so many universities, mostly in the Northeast, obviously, because that's where we are, so it's easier for them to get there. New York Genome Center, Nano, Genomics, Illumia, Perkin-Elmer, Cold Spring Harbor Laboratory, Biorad Labs, so many others. So it was a lot of different people. Good mix. There was a lot of people from JAX, obviously, but yeah, there was quite a few different representations of companies and people and universities. So, but as I said before, there are many other conferences coming up in Jackson, our different Jackson locations, and there's 2 more in October and 4 in November. So you can check out the full listing online at courses.jax.org/event listing. There's a hyphen with the event listings between that. But I'll put the link in the show notes on the website, dnapodcast.com, so you can check it out there. Or if you're listening on iTunes, then it's right in the description there. You don't even have to go to the website. It saves you a step. But to name a few happening in Farmington, there's the Cancer Genetics Management in the Primary Care Setting. That's November 7th through the 8th. There's a workshop on 3D genome map technology, chromosome interaction analysis by paired end tag sequences. Ooh, big words here. November 9th to the 14th. And Immunogenomic Symposium, November 13th to the 14th. They were more heavily on the marketing on that last one. But yeah, those are all happening actually in Farmington, here in Connecticut. So you can check that out online. Again, that's courses.jax.org/event-listings. And I'll put that in the show notes for you. And we have about 5 minutes. So I'm going to go over the news story that I kind of hinted at before. And that's about the BRCA2 gene. So October is Breast Cancer Awareness Month, so I thought a news story about the BRCA gene would be fitting. And the BRCA gene is associated with breast cancer. That's why it would be fitting. So I've talked about the BRCA genes before on the show and discussing how the BRCA genes are associated with ovarian cancer as well as breast cancer. And this was during the news story about Angelina Jolie and her being very open about getting a double mastectomy and her impact on people getting their own genetic testing done. And there was research that showed, months after she had made her announcement that genetic testing had skyrocketed, then leveled off. But it just goes to show that, when people are in the news, especially celebrities talking about it, that people are more aware of it and asking healthcare professionals about it, inquiring for themselves. But we're talking about the BRCA2 gene here, and UK researchers have taken their first pictures of this protein, the BRCA2 protein. And this is the first step of discovering more about the structure and mechanism of the protein by looking at it. It's kind of like how DNA worked, how Rosalind Franklin was able to take a picture of the DNA, and from that we learned so much. So it's kind of comparing it to that if we're taking picture, we're kind of starting from there to learn more about the structure and mechanism of the protein. And once we do, treatments that target the gene mutations of the BRCA2 gene will be easier to work on because we'll know more, and it's little bit easier to figure out what's going wrong with it in mutations and how we can fix it to make it work properly. So before this study, the current research was that the BRCA2 gene was associated with DNA repair. So if DNA needs repairment, then BRCA2 was somehow influenced or part of this process. Now, if there's a mutation in this gene, then the repair on DNA is hindered. So if the BRCA2 gene isn't working, then repair on DNA isn't working either. It's causation right there. And researchers now have revealed more information, and they found that the BRCA2 proteins work in pairs, and each works with another protein called RAD51. And so each of these proteins works with RAD51 to repair the DNA. So they're kind of partners in crime here, repairing the DNA. And these RAD51 proteins form filaments, kind of little pieces, and find the matching DNA strands to repair it. If there's a broken part of the DNA, it will kind of make that piece and fit it in there so that it works again. And so these RAD51 proteins work in opposite directions, so they can both work on both sides of the DNA. And it was discovered that the BRCA2 proteins have a larger role in actually forming these filaments, these pieces that RAD51 is forming to fix DNA. And Professor Zhang of the Department of Medicine at Imperial College London led the research team that worked on this project. And I'm going to share a little quote that he said. He said, quote, "This study improves our understanding of a fundamental cause of cancer. It's our first view of how the protein looks and how it works. And it gives us a platform to design new experiments to probe its mechanism in greater detail. Once we have added more detail to the picture, we can design ways to correct defects in BRCA2 and help cells repair DNA more effectively to prevent cancer. We can also think about how to make the repair process less effective in cancer cells so they die. So the cancer cells die and our cells don't die." So they published their results in the journal Nature Structural and Molecular Biology. So this is really interesting that if they are able to really understand what's happening, that what's going wrong in BRCA2 mutations, and then figuring out a way to fix those, then we wouldn't have breast cancer, pretty much. I mean, it would at least be less, and it would help whatever's going wrong. If we can fix that, then it's all good. And so at least breast cancer would go down, and that's kind of, that would be a major advancement, and that would be a huge breakthrough. But just some stats to throw at you regarding the relationship between the BRCA2 gene and breast cancer. Around 45% of women who have a BRCA2 gene mutation will develop breast cancer by the time they're 70 years old, and this number can be compared with 12% of women in the general population of getting breast cancer. Similar stats occur in ovarian cancer. 11 to 17% of women with BRCA2 gene mutation will get ovarian cancer, and only 1.4% of women in the general population will have ovarian cancer. So, interesting stuff. Breast Cancer Awareness Month. I'm sure you've heard of it, and it's kind of all over the news. But that's all the time I have today for DNA Today. And you can tune in next week, Mondays at 1 p.m., and every Monday at 1 p.m. to learn more about genetics. You can follow the show on Twitter at DNA Podcast, and shoot me email questions at info@dnapodcast.com if you have any. And follow Jackson Labs at Jackson Labs on Twitter. And join me next week, Monday at 1 p.m. and every Monday at 1 p.m. to learn and discover new advances in the world of genetics.